The broadcast is now starting. All attendees are in listen-only mode.
Good afternoon, ladies and gentlemen. Welcome to today's webinar featuring our latest software release called QuiC, a QC analysis tool that is fast, easy to set up, and produces intuitive readouts for your quality control measurements of your LC-MS setup. Today with me here is Lukas Reiter, Head of Research and Development Biognosys. I am Florian Marti, Product Support Manager, and will present you today's webinar. First, we're going to give you a little introduction into the life cycle of a software tool in Biognosys. Then we're going to talk why good quality control is essential to perform proteomics measurements. Then we look into some quick facts. Then we look at the tool in action, how it works for mainly DIA, DDA, MRM, and PRM measurements. And in the end, we will have a short Q&A session where Lukas will discuss some of the questions you might have considering our tool. The software tool QuiC presented today is a prime example how Biognosys actually works in its core between the R&D department, the contract research department, and the integrated workflows or the product department. QuiC was initially discussed with some of our product customers that said that they're lacking some good quality control software, and we then went back to our R&D department and asked Lukas and his team if they can actually develop such a tool. What they did, as you can see today, they developed one of these tools, which we are now giving back to our product customers, but we are also using it in our contract research projects to show our customers how their data looks and to judge the data based on QC parameters. Good quality control, or QC, is an elemental process of quality assurance in proteomics. In general, QC readouts can provide you valuable information on sample preparation, on the liquid chromatography part, and also on the mass spectrometer, and gives you thus readouts how well your sample preparation worked, or if your mass spectrometer actually worked optimal, or if you have any trends in your mass spectrometer over a period of acquisition. And generally, QC tools, they work on different levels, such as the mass spectrometer, the spectra, or the bioinformatics part. Some of the tools that are available are also working on multiple parts, but so far, to our knowledge, there was no QC tool that actually summarized all the different parts for all different workflows. Good QC can be done on different levels, and the first and most known level on which QC is done is when you're measuring an experiment. You generally have a quality control sample at the beginning of your acquisition. Then you have one in the middle and one at the end. And with these samples, you're basically determining how your LC-MS setup performed over time during the acquisition of all the runs in between that are used for the analysis of the experiment. What is then done, generally these runs are fed into a QC tool of choice, or one of the different tools that are available. And if everything went well with these QC tools, the user generally accepts the values and thus goes on with the analysis of the data. But doing QC on dedicated QC runs is not the only way we think QC can be done. Another way to do QC is also on all runs of a certain experiment to identify outliers due to either chromatographic performance issues or to mass spectrometric issues. And as you can see here depicted, we have again the same runs, so all the experiment-specific runs, and all these runs are fed into the QC analysis pipeline to distinguish which runs are good, as you can see here, and which runs are bad and should be therefore excluded from the data analysis. As I mentioned before, when we look at the current QC tool situation, we see that quality control is generally done at different levels, such as sample preparation, then at the liquid chromatography part, and also the mass spectrometer part. Further, QC can be done on the spectra or on the bioinformatics pipeline using the identifications or quantifications readouts from the different methods. Normally, different tools are used for different read levels, as I've shown you on the slide before. And moreover, different tools exist for different mass spectrometric vendors. Apart from that, also different tools exist for different proteomic workflows. And no tool, to our knowledge, up to date is actually summarizing all major workflows from all major mass spec vendors. And this is the step where we actually talk to our customer, and we wanted to provide the community with a tool that supports the major proteomic workflows and also supports all major vendors. As you're attending this webinar, you all know that we have released such a tool. It's called QuiC, for a fast and easy to use QC analysis tool. And as all of our software releases, we gave QuiC a software name. And in tribute to Jesse Owens, we have named QuiC 1.0 as Owens. It reflects actually the name of Jesse Owens in a lot of ways. So as you all know, Jesse Owens was one of the most famous athletes in track and field history, which is reflected in QuiC's name for being fast, but also easy to use, and not only being fast in the analysis, but also being fast in how it was developed. The idea of releasing a QC analysis tool was already existing for quite some time within Biognosys, but there was never really an urge to program such a tool. Then in September 2016, we talked at a conference with different customers, and they all requested such a easy-to-use, intuitive QC tool. We then went back to our R&D department, and they started working on a QC tool. And at the MSACL US 2017 in January, we released a test version to our customers. Last week, we have then officially released QuiC 1.0. As I said in the introduction, QuiC was very fast in its development, but it's also very fast in its run. As you can see here, a QuiC analysis takes about 2 to 8 minutes, depending on what kind of run you're analyzing, on a low-end hardware like a common laptop. Main features other than being fast is that QuiC is easy to set up. It is intuitive to work with. It is also very robust and incorporates corruption checks for your files from different vendors. It supports, as I said, multiple vendors. At the moment we have full support for Sciex and Thermo Fisher, and are of course in the process of adding more and more vendors over time. It supports all major workflows such as DDA or shotgun proteomics, then the data independent acquisition, MRM and PRM. It allows the tool allows the real-time quality control monitoring of specific QC samples, but also enables real-time monitoring of individual runs from large-scale experiments, and thus can save the user some analysis time, because the tool enables you to objectively exclude certain runs from downstream analysis. In the next couple of minutes, we're going to look a little bit into QuiC and how it is built up and how it is work. After installing QuiC and receiving your license, you are ready to go and perform QC analysis. QuiC allows you to either add files manually and by browsing to the respective folders, but it also gives you the option to do the real-time folder monitoring to perform on-the-fly QC analysis every time a new file is stored in this folder. This is especially valuable when judging the mass spectrometer over time, when you don't need to wait for a long time until new files are actually in the folder again, because of the fast processing speed of QuiC. The information within QuiC is logically stored based on the instrument, then on the workflow, then the experimental date, and last but not least the individual runs. Parameters of the mass spec, the IRT peptides, some general QC features, as well as discovery and advanced shotgun proteomics measurements are monitored. As with all our softwares, there are tooltips to help understand each parameter. By mouse overing the names, you get an information what is actually calculated. QuiC also allows you to filter the different data, and you can browse through the data. As with Spectronaut or SpectroDive, whenever you change the level of the data on the left side in the history, the plots on the right side, they dynamically change based on the data level. A first plot that we look here at is a quick plot for the mass accuracy. So we're looking at the mass spectrometer. Here depicted are individual QC samples that we use internally for our projects. The QC samples are C. elegans lysates that are measured in repetitive intervals within the projects or in between different projects. Each box plot here corresponds to one run, and the runs are sorted by acquisition date. As I said, plotted is the MS1 accuracy, and each plot equals one measurement. As you can see here, if we look from left to right, we see that over time the mass spectrometer actually starts to have a decreased mass accuracy, and as depicted in red, we can see representative points when calibration was due. So as you can see here, QuiC really helps you to process your mass spectrometer and to helps you to define at what time point calibration or cleaning or tuning is due. As I said, the plots are intuitive and they change dynamically based on the level of data we look at. So here's the same plot as shown before again. Now on the date level, and you can see when the calibration was done in the first, and then how the mass accuracy behaves over time. On the right, we have the same plot now shown on the individual run compared to the entire level. Some more plots that QuiC is displaying are, for example, plots on the retention time on the left and on the IRT on the right to monitor the performance of your chromatographic setup. And already looking at these plots, you can see why the IRT plots are actually the better measurement to determine whether chromatography was stable or not. If the user would look at the retention time only, you would probably think that here after the second run something with the chromatography went wrong. But if we look at the IRT, we see that the chromatography was very stable, or in other words, the main difference leading to the two different plots here is just a different gradient length. What QuiC also allows you is to display certain features together, as depicted here in blue, by simply clicking, enabling them together, and then plotting both values here. Again, the retention time versus the IRT together, and you can basically see that everything was stable and chromatography worked very well. This already brings us to the end of this very fast presentation about our QC analysis tool. As I said, QuiC is fast, it's easy to set up and intuitive to work with. It supports multiple vendors, at the moment Sciex and Thermo Fisher instruments. It supports all major workflows from DDA, DIA, MRM and PRM workflows. It allows you to real-time QC monitor of specific QC samples, or it also allows you to analyze all your MS files from a certain experiment to perform QC analysis and to determine which samples can be excluded. Last but not least, QuiC is free of charge. It only requires the IRT peptides to perform certain analysis. If you wish to request your own license of QuiC, just send us an email at support@biognosys.com. Lucas and me will now take any questions that you have considering QuiC and its features. Okay, we see the first questions coming in. So the first question was, are also older mass spectrometers supported like Thermo or the Orbitrap XL? Yes, so QuiC is QuiC analyzes all mass spectrometers from Thermo and Sciex without any restrictions on age. So there's like the entire platform is supported. The next question, is SBS-MS3 TMT-like data supported, and is it necessary to have the IRT peptides in there? In other words, which features will I lose without it?
Okay, so I can take this question. The IRT peptides are needed in general for the QuiC tool because it will not run any analysis if the IRT peptides were not in the sample. With regards to TMT and MS2-3, it is possible to use such samples with QuiC. What is needed basically is a spectral library including TMT, but such a library can be generated using Spectronaut. So spectral libraries can be generated in Spectronaut and can be imported into QuiC, but you will not get MS2-3 level readouts at the moment. You will only get MS2-2 level readouts. And there will also be no TMT specific readouts at the moment.
The next question is, can several instruments of the same type be monitored separately from the same QuiC instance?
So that's a good question. What we basically do is, it's very simple. Using the vendor API, we read out the specific instrument, the unique instrument ID from the instrument, and this is then being used in the software to display all the different instruments. So yes, one can use several instruments of the same vendor and of the same type within QuiC. However, if the mass spec vendors do not fill this field with a unique ID, then the API will report either a blank string or something like this, and our software will also not be able to distinguish the instrument. So it's basically down to the mass spectrometer vendors to really fill this unique field in the API that is being intended to be used for a unique ID of the instrument.
The next question, are only the IRT peptides analyzed, and what is the background library for only for DIA and not for DDA? So, as we mentioned before, QuiC is doing QC analysis on two parts. So the first part is the IRT peptides for all workflows that QuiC is able to do the analysis, and on top of that, QuiC is also doing a QC analysis on a pre-specified library for DIA and DDA. This library comes in the kit file and per default is set as a HeLa background. But as Lucas mentioned, the user has the freedom to change this library in the settings perspective to any other library he wants to use, and this can easily be done by generating a spectral library in Spectronaut, or if you don't have a copy of Spectronaut on your computer, you can also contact us at support@biognosys.com and we can generate or we can provide you with such a background library. Then the next question is about the QC standard mix and what the QC standard mix is. So the QC standard mix is the IRT peptides that are non-naturally chemically synthesized peptides that were first introduced for scheduled MRM and were later adopted for the analysis of DIA or SWAF data. The IRT peptides can be ordered either via our online shop or they can also be ordered by sending an email to order@biognosys.com. The next question is, on what level do you recommend performing your QC analysis?
Okay, here at Biognosys we perform QC on several levels, as Florian already indicated before. We typically perform a quality control run before and after instrument maintenance. We also perform quality control in special QC runs within an experiment, especially if it's a very large experiment. If it's dozens or hundreds of runs, we do that. And we also perform quality control in every individual quantitative run, because we have the RH peptide spiked into every run. So we perform a sort of a 3-tier quality control, which gives us very, provides us with very robust instrument performance and always performance we'd like to have. And we are also quick to intervene if something is not going correct in measurement.
The next question is about the costs that are added per run by using QuiC. So as QuiC is a free tool to use, the software does not add any costs. The IRT peptides spiked into every run at, according to the manufacturer's instructions, are adding about 0.05% of costs to each run. This is, if you translate it into dollar values, it's about 20 cents per run. And the next question is if we could get a copy of your presentation afterwards. As with all of our webinars, this webinar is recorded. A recording will be provided, and I'm also happy to forward you the presentation for your own, to review it at your own pace. Are there any more questions? It seems that all the questions have been answered. We thank you very much for your attention, and we wish you a very nice day.
Thank you very much. Have a nice day.